sexual reproduction is the opposite of asexual. It means that we need two gametes, a male gamete, for example, like a sperm in animals or a pollen in plants, and a female gamete. They have the same name in animals and plants, which are the ova or ovum or ovule or excel, as you like. But here, when these two gametes fuse together, because every one of them contains n number of chromosomes, so they give me a 2n, or they give me a zygote. And I explained this yesterday about the fusion of the nuclei, the nucleus only, of the male and the female gametes. They give me 2n nuclei, zygote, sorry, and this zygote, because it carries chromosomes from the father and from the mother, so this zygote is carrying a collection of genes, different genes, so we say this genetic or genetically variants or genetic variation. We can call it like this because this description is very important. In the mitosis, there was no genetic variation because it was one cell that divided into two. So the daughter cells, they are genetically identical to their mother cell, exactly like a copy and paste. But what about this? No, because the zygote is carrying information from two different organisms. So it carries the information from the male with the information of the female, making this zygote not identical to the father or to the mother, but gathering information from both of them. Now, what are the advantages of this sexual reproduction? Of course, genetic variation, because as I said yesterday, that this is a great advantage, because if something happened or changed in the surrounding environment, these zygotes with genetic variation can face these changes and can survive. For example, like if something happened about the climate, or there are some plants, for example, that grow in cold regions or in temperate regions or whatever. So if the climate changed, the ones with genetic variation are going to face these changes, and they are strong enough to survive in these environmental variation. In asexual reproduction, no, they do not carry different genes. That's why they will not be able to survive if the climate, for example, changed. So if there is any change in the environment, genetic variation will allow these organisms to face these changes in order to be able to survive, to reproduce, and so on. So what are this is the advantage, of course. What are the disadvantages? Disadvantages here, for example, we say that the sexual reproduction is more risky than the asexual reproduction. Why? Because every male or female, they must find a mate. And I mentioned this word yesterday, finding a mate. Sometimes they do not find a mate. Sometimes when there is a collection of animals, for example, most of them are males and only one female. What about the rest of the males? They are not going to find a female in order to mate. There is difficulty sometimes in finding a mate. And another disadvantage is that sexual reproduction requires sexual organs. You're going to say, does any individual does not have sexual organs? No, they have, but they have to wait till these sexual organs mature or they become effective in order to sexually reproduce. Sometimes there are certain mutations that we are going to learn later. They change the shape of the sexual organs of the animals, for example. This is mostly noticed in animals or plants. I'm going to mention these later on. We are going to learn something about mutation of these sexual organs in details later on. But these are the disadvantages. Type. Of course, I gave you yesterday a complete idea about the gametes, and they are formed of the gonads, and the number of the chromosomes found in the gametes. But let's now go to meiosis, or the reduction division, because the number of chromosomes is reduced to the half. Just a second. No. The number of chromosomes is reduced to the half, and we can call it meiotic division. They are all correct. But here I say we are going to describe this as a reduction cell division in which that occur only in the sex organs. Yani if I have, this is the testis, not all of the testis cells are going to give me sperms, but some cells only, they are their function is to divide by mitosis, while the rest of the testis, they divide normally by mitosis. Only a part of it, the cells will divide by meiosis in order to give sperms. And the same happens in the female. If I have an ovary, not all the ovary will give me ova. A part of it only in the material of the ovary is going to divide by meiosis to give me the ova. This is plural, of course. As I told you yesterday, that these organs, whether the testis or the ovaries, they are called the gonads. But this means that some cells of the gonads are responsible for producing sperms and ova, in male and female, of course. But what about in plants? In the flowering plants, there are as well special cells that are going to that will evolve to give the gametes. Of course, if I have a part of the cells of the plant that are going to give me the male gametes, we call them the microspores. These are certain cells on the male parts of the flower. They are going to divide by meiosis to give me pollens or pollen grains. While on the female part, they are called the megaspores. And the megaspores, they are going to give me the female gametes or the ova. But why micro? Because the pollens, they are microscopic. But why mega? Mega means very large. Of course, if I compare the ova, the size of the ova to the size of the pollens are very minute, and the mega and the ova, they are very large if compared to the pollens. But that's why the parts of the cells in the flowering plant that will give me pollens, they are called microspores, while the others are called megaspores. And of course, whether in animals or in plants, the fusion of the nuclei of these gametes will result in variation, or genetic variation. Remember all the time that the sexual reproduction results in this genetic variation. Right. What happens to the chromosomes during meiosis? I'm going, of course, to explain this later on, but I'm going just to give you titles now. Meiosis is a kind of cell division, so it is preceded by an interphase. Like what I explained yesterday, is that the cell becomes big in size, bigger. It performs, it takes in many materials. It performs more and more metabolic reactions, produces more energy. The cell is ready for division, وبيحصل replication of DNA and so on, doubling of the organelles, exactly like mitosis. It is preceded by an interphase. But there is a certain thing that happens to the chromosomes which is called crossing over. to the chromosomes in meiosis only. Crossing over, it has another name, recombination. And this crossing over, from its name, it means that because the cells they have, of the nucleus, it has a pair of chromosomes. One is from the father, the other one is from the mother, like this. So the crossing over will happen between these two homologous chromosomes. I'm going, of course, to explain this later on, and this crossing over results in what we know as, again, or adds to the genetic variation. يبقى الgenetic variation here resulted from two factors. Different source of chromosomes, one from the father, one from the mother, and the crossing over. I'm going to speak about the causes of genetic variation after I finish explaining meiosis. Now let me put a picture to show you meiosis and how it differs from mitosis. Now, if you can see here, طبعا this drawing does not have the interphase, because we already know interphase. It is preceded by interphase. As you can see from the picture, that meiosis is divided on two stages. We call these stages meiosis 1, or the first stage of meiosis, and we write 1 in Roman numbers. Yes, in the book they wrote it like digits like this, but this is not correct. I always like to use the Roman numbers where scientifically they use the Roman numbers, while on the other side this is meiosis 2. But let's go to meiosis 1, and let me remind you by the pro-interphase when I said there is doubling of chromosomes or doubling of DNA or replication. And as I said yesterday, replication of DNA results in doubling of the nucleus, in the chromosomes, of course, because the chromosomes, they are formed of DNA mainly. If the cell started before doubling, I'm going to give you طبعا an example. Like I mentioned yesterday, we have, for example, 46 chromosomes. I'm not going to put them all inside on this drawing. I'm going to use only 2 chromosomes as an example. The rest of the chromosomes will act the same. So 2 chromosomes, when they double in interphase, they are going to be 4 chromosomes. Each one will make a copy of itself in this interphase. Here are the 4 chromosomes. Every one is making a copy of itself. This is the number of chromosomes after doubling. If in human beings, normally in the 2N cells, we have 46 chromosomes, so after doubling, they are 92. Here, this picture represents the 92 chromosomes. Now, the same as I said yesterday about mitosis, we have the same phases. The first phase is called prophase 1. Why 1? Because it belongs to the first stage of meiosis. In prophase 1, certain events are going to happen. The centrioles, they are going to migrate each to one pole of the cell. Nucleolus, it's not drawn at all, it disappears, and the cell membrane, or the nuclear envelope disappears. These are the events that happen in prophase 1. And in prophase 1 as well, there is crossing over that I'm going to explain later. Prophase 1 is the longest phase because several events happen in it, plus the crossing over that elongate or lengthens the prophase 1 more and more. After the nuclear envelope and nucleolus disappear, while centrioles, they reach the poles of the cell. So the next phase is going to start, which is metaphase. Again here, metaphase means lining up of chromosomes in the equator. But what's the difference between the metaphase of meiosis and metaphase of mitosis? Let me remind you by the metaphase in mitosis that I explained yesterday. This was the cell membrane, and chromosomes, they lined up one after the other like this. Okay, let it be 4. Ahsan. I'm going to widen this. Here we have 4 chromosomes that are lining up, and we knew yesterday that the centrioles sending the spindle fibers to be attached to the centrosomes of each chromosome. So when the spindle fibers shorten or retract, they are going to separate the chromosomes from each other, the chromatids, sorry, chromatids from each other like this. I explained this yesterday. And the chromatids are going to be separated. This is mitosis. But let's look at meiosis. Here in mitosis, all the chromosomes lined up in the equator. But look here, the chromosomes, they lined up 2 by 2, not 1 by 1 like this. Each chromosome is going to line up with its homologue or with its copy. They are going to stand like this or to line up in the equator. But how are the spindle fibers going to be attached? They are going to be attached, this group will be attached to these chromosomes, and this group will be attached to the other chromosomes. And let me draw this. Now suppose this is the cell membrane, and I have here 2 chromosomes, 2 by 2 like this, and spindle fibers are attached to them like this. Elongate and attach to them. But what if the spindle fibers shortened? Yeah, Mariam, are they going to separate the chromatids or the whole chromosomes from each other?
If the spindle fibers shorten?
Yes. Yani in mitosis, they separated the chromatids. What about in meiosis?
They're going to separate the chromosomes from each other because they are lined up 2 by 2.
Yes, very good. Yani look here at mitosis. The spindle fibers were attached to the centromere of the same chromosome and from the other side. So when they shorten, they are going to separate the chromatids from each other, as if I have a chromosome like this, and then the spindle fibers separated the chromatids from each other because they were held here, and every spindle is pulling to an opposite side, so the chromatids separated. But here I don't have spindle fibers. They are not attached to chromatids of the same chromosome. No, each spindle is attached to a separate chromosome. When the spindle fibers retract or they shorten, they are going to pull the whole chromosome with them, not half of it. Is this understood, Warda and Salma?
Yes.
When the spindle fibers shorten, this phase is called anaphase 1, the same names. Now here, as you can see, that the cell started, or the cell membrane started to constrict from both sides. So with further separation, there is further constriction until the 2 chromosomes here, they are completely separated from the other 2, the nuclear envelope is going to reform. Centrioles, they return back to their place, and the spindle fibers nearly disappear, where cell membrane will keep on constricting like this. But the two cells are not separated from each other. Yani here in meiosis 1, there is no cytokinesis. What's the meaning of no cytokinesis? Means no separation between the two cells. In mitosis, the two cells completely separated from each other. But here in meiosis 1, there is no cytokinesis, means that the two cells, they keep attached to each other. Why? Because there is another stage for meiosis. We need these cells to keep attached to each other. And here, this telophase 1 or of meiosis 1 is considered as prophase for meiosis 2, as if it is the prophase of meiosis 2 or prophase 2. Here the cell in telophase 1, two cells that did not separate completely, they are going again to lose their nuclear envelope, okay? And the spindle fibers, they are going to lengthen again. And here, look at this cell and look at this cell. These are these. Now, look at the chromosomes. They lined up in the equator. And the spindle fibers, they attach to the centromeres of the same chromosome. Now, let me again remind you what happened in mitosis, but with one cell only. In the chromosomes lined up in the equator, and here are the centrioles attaching spindle fibers to the centromere of the same chromosome. Tab Salma, if these spindle fibers shorten, what will happen to the chromosome? Will the chromatids be separated or not?
Yes, they'll be separated.
Of course, طبعا. طيب, the same thing happens here. When these spindle fibers shorten, they are going to separate the chromatids from each other and for the other cell as well. So this stage is exactly mitosis. That's why I said yesterday that mitosis is a part of sexual reproduction. It is meiosis 2, or the second stage of meiosis is mitosis. But let's continue. Here I have mitosis, but in two cells, not only in one cell. Here, this is anaphase 2, where during a shortening of the spindle fibers, the cell membrane constricts or indents to the inside. But during further constriction, the nuclear membrane is going to reform, and the two cells are going to separate completely from each other. And not only the two cells, there is a separation here as well. So these are going to result in 4 cells. Okay, type. We started by 1 cell. It was 2N. And I give you an example by having only 2 chromosomes before interphase. Okay, remember what I said at the beginning. I'm going to start with only 1 cell, 2N, one of the gonad cells of the testis, of the ovary, of the anther, of the ovary of the plant. But each cell is a body cell. Normal body cell means 2N. And I took only 2 chromosomes as an example. And this is before replication. But look here at the produced cells or the daughter cells. Warda, How many chromosomes are found in each cell? How many?
One.
One.
Chromosome.
Yes. So the daughter cells, they were how many? 4 daughter cells. Each one is having half the number of the chromosomes we started with. Okay. What's this? This is the returning back of everything again back to normal. Remember when I said in the chromosomes, they do not appear except when the DNA condenses. Here, the DNA is going to decondense. It means that it will return back as very long threads that are translucent and they will not take the stain. The chromosomes, they lose their shape خلاص because the DNA decondenses, because I said yesterday that the DNA is in the form of very long threads that cannot be seen. But when the cell starts or is about to divide, it starts to condense when I said coiling and supercoiling. So they become thicker, they become shorter, they become attached to the histone proteins, and they allow the chromatid to be formed. Or in other words, if 2 chromatids or sister chromatids, they come in contact with each other at the centromere, this will form the chromosome and they will appear. Now here, I'm returning them back to their actual shape, decondensed chromosomes. They do not appear, they return back, the DNA return back as a thread, a very long thread, uncoiled يعني. Okay? This is what happens in meiosis, and as you can see that meiosis 2 is just simple mitosis. But here in meiosis, if I want to compare between them بقى, in mitosis, and in meiosis. In mitosis, there was a body cell containing 2n chromosomes, or the total number of chromosomes. And when the cell divides, it gave me 2 daughter cells having the same number of chromosomes and the same types and everything, genetically identical. Is there any genetic variation? No genetic variation. Why? Because it was one cell that made a copy from itself 2 times or twice of itself, an exact copy. No genetic variation. Nothing is changed in its genetic composition. Now, what about the meiosis? Meiosis, it was a body cell with 2n, resulted in 4 N cells or haploid, having half the number of chromosomes, and these are not genetically identical. And of course, there is variation or genetic variation. Type. I said before that there is something called the genetic variation is very important in a very great advantage for meiosis. Let's go to this genetic variation. And what are the causes of genetic variation? Yes, I said that there are 2 causes, but I'm going to add another one now. The first cause, I'm not going to write it. It means that one chromosome is coming from the father and the other one from the mother, or in other words, from the male and the female. Yes, we know this. But what about things that you have to know, a new information, something called the independent, these are the causes, independent assortment. What's the meaning of assortment? Assortment means choice or choosing. Independent means random. So independent assortment means a random choice. Now, what does this mean? It means that if I have, for example, this is a male or yani a human male, and this is a human female. Suppose I have here 23 طبعا sperm and ovum. Suppose I have here 23 chromosomes and I have here 23 chromosomes. So الزيجت أو this gamete, it has chromosomes from its father or mother. يعني this gamete can contain, I'm going to say it and then I'm going to explain it. 0 or 23 chromosomes from both parents. But what does this mean? Look at the parents of this male. They were 23 in a sperm and 23 in an egg cell or an ovum. And when these 23 and 23 fuse together, or the nuclei fuse together, they got this male. But what about the sperms of his father? The sperms of the sperms of the father, they are having 23 chromosomes from their parent type. This sperm can carry either no chromosomes at all from its father and 23, the whole 23 from the mother. Sometimes the sperm can carry, for example, 2 chromosomes from the father and 21 chromosomes from the mother. Sometimes they carry 23 from the father and nothing from the mother, 0. It means that when the sperm is formed, it is not choosing which chromosomes to form it. It's not choosing. Finally, it must have 23 chromosomes. But is it choosing which chromosomes are going to find inside the sperm? Of course not. That's why this process occurs randomly, or there is nothing is chosen, just a random collection of 23 chromosomes. Maybe this sperm gathers some information from the father and some from the mother. Okay, maybe no information from the father and some from and the rest from the mother, and that's it. This is a random collecting of chromosomes inside the sperm. And the same happens to the female ovum. Maybe it contains a number of chromosomes from the male parent and the rest from the female parent. That's why I said that each gamete contains from 0 to 23 chromosomes from one or both parents. And of course, this is because the distribution of chromosomes in the gametes is a random process. No gamete is choosing which chromosomes to be inside it. الفاينل يبقى 23 chromosomes, but coming from where? This is not something calculated or studied. No, it is a random choice, a random process. But this random or independent assortment results in genetic variation. But what about the other cause of genetic variation? It is crossing over. And this crossing over, it happens in prophase 1 of meiosis, not mitosis. Mitosis does not have a crossing over. Crossing over is in prophase 1 only. By the way, before I start this, here, this random choice may result in about, when they studied this, they found out that there are 8 million potential genetic combinations, like probabilities. Remember when I was explaining the proteins and I said there are 20 amino acids, and according to their number, their sequence, their kind, we have the whole protein, and I give an example bar by amino acid A linked to A linked to A, and it's changing. But these are called the probabilities or the potential combination of amino acids. Here is the same. I have different probabilities for collecting genetic information from both parents on the gametes. When they studied something like that, they were brought to a very amusing result, that because of this random or independent assortment, there might be 8 million potential genetic combination within a sperm and within an ovum. ممكن يبقى probabilities they reach 8 million probabilities. So what about, it means that the gametes, they have great variations in the same person. So what is this? Yes, Mariam.
I understand the concept of the combinations, بس أنا السؤال هنا who are the parents؟ مش هي it's usually one cell that divides into 4 to make the gametes.
I'm explaining that the chromosomes that are found in the gametes, the chromosomes, they are not, ما هو الgamete is coming from a person, and this person is coming from the parents. Okay, if this person مثلا 1 أو 2, person 2, it has a parent, male and female. So in person 2, these are going to give chromosomes to the sperm of person 1, or the ovum, of course. Okay, the chromosomes that are found in this sperm, or if it's a female يبقى من الأوفم, عندي actually they are coming from their parents. Okay, chromosomes that are coming from the parents are assembled in the sperm randomly, or they are put in the sperm randomly, not by choosing some chromosomes in particular to be here and the others not to be here. For sometimes this man is going to carry no information on the sperm from his father, 0, but the rest, because we have 23 from the mother, all of them. Suppose this sperm contains 4 chromosomes from the father and 19 from the mother. Okay, and the opposite, maybe 19 from the father and 4 from the mother. The final in this sperm must have 23 chromosomes. Okay, it's not a must to be, for example, 11 from the father and 12 from the mother. No, no, it's something randomly. Some are coming from the father, some from the mother, and maybe nothing from the father and all from the mother, maybe nothing from the mother and all from the father. Okay, what's more important is that the final result is 23. Coming from which parent? This is a random process. Random
Doctor?
process. Yes, yes, Anna.
So this only affects sperm بتاع offspring, صح? So it doesn't have anything to do with the phenotype.
لا
was more leaning towards
genotype. يا سلمى, this is genotype. بس later on بقى لما الزيجة يتعمل والoffspring يطلع, طبعا the genotype will affect the phenotype. مش كده? As we learned before. وباي ذا واي يا سلمى, this is not only for animals, ده for all living organisms, للplants كمان.
Okay.
طيب, let's see what is this crossing over. I'm going to put here a picture to show you what happens exactly. And this is again in phase 1, not 2. We're not in mitosis, only in meiosis. Type. I'm going to zoom this in a bit to show you the colors. They are very faint. I think you can see the chromosomes on top. Here I have one chromosome, and this is its homologue. if this chromosome is coming from the father or from the male, if I'm speaking about plants مثلا, and this is coming from the mother. Now, as you can see, that these are the homologous chromosomes. What's the meaning of homologous? Same length, same number of genes, same kind of genes, same loci of the genes, and the gene is found in the same place on the other chromosome as well. Right. Both chromosomes, they give me how many chromatids, ya Warda? How many chromatids are here for both?
I'm sorry, can you repeat? How many chromatids for both?
Yes.
4.
4. That's why we call this shape a tetrad, because tetra means 4. 4 chromatids, okay? A tetrad. Here I have 4 chromatids of homologous chromosomes. The ones near each other are called the neighbor chromatids. Crossing over is going to happen in the neighbor chromatids only, not in the distant chromatids. The neighbor chromatids are the 2 chromatids that are near to each other, one of the father and the other one of the mother. They are going to approach each other and see, they crossed each other. Now, crossing here, it gave me the shape of a letter X. This letter X is called a chiasma. Whenever you find crossing كده between 2 structures, this is called a chiasma. And you have a chiasma in our brain, but not, of course, of chromosomes. Anything that gives the shape of a letter X, so this is called a chiasma على طول. Type. As you can see here, that this chiasma allowed the genes that were found on the pink chromatid to be transferred to the green chromatid. And the genes of the green chromatid, they were transferred to the pink chromatid. This means that both chromosomes, they exchanged their genes. Taban, this is going to add again to genetic variation, more genetic variation. Yani it's not enough that one chromosome is coming from the father and the other one is coming from the mother. No, crossing over is going to add to genetic variation. It's not only enough independent assortment. Crossing over is going to add more and more genetic variation. Like, is chiasma only happening here? No, no, no. Chiasma may happen here or here or here or here, anywhere on the neighboring chromatids. Will only be one chiasma? No, they can be many crossing overs here, and we call them the chiasmata. This is the plural. Chiasma is only one, chiasmata are all of them. Yani if I can draw it, I'm never able to draw it, but I will try. This is, for example, this is the centromere. There is chiasma like this, for example. And this is the other chromosome, like this, for example. Oh, many chiasmata, 1, 2, 3, 4, 5. Yani el-chiasma, it happens either one chiasma or many along the whole length of the chromatids. Of course, when there is more chiasmata, then there is more and more genetic variation because there is more and more exchange of genes. This process will result, look here, I have here, let me draw this picture up. I hope إن أنا أقدر أطلعها فوق. No, never. It returns back. Look at this. When there is meiosis and the chromatids are going to separate in order to give me يعني طبعا الchromosomes, daughter chromosomes at the end. For this one here, this one without any variation here, but these are going to be found like this. يعني exchange genes, they are going to be transferred to the gametes, of course. And the ones, the chromosomes that have crossing over, we call them recombinants or recombinant chromosomes, because the crossing over has another name, recombination. And look at the word re, means again another combination. So I have here the primary combination, while in the neighboring chromatids I have another recombination, a recombination to the original. So the resulting cells are called recombinants. So here I have normal cells, يعني original, like the original chromosomes, because they do not have crossing over. And this one, while these two are recombinants because they had crossing over. This is another cause for genetic variation, or it adds for genetic variation. If you have any question here before I start the most beautiful part in this chapter, where I am serious, it's very, very nice. You have any question in what I explained previously? Nobody's telling me. It means that if you do not answer, I will consider that you do not understand. If you stop talking, you do not answer me, I'm going to stop explaining as well. Let us remain in silence. Any question?
No questions.
Yes. Let me hear your voice. To be sure I'm not alone. Okay. Now let's go to the formation of gametes. How the gametes are formed, and this is called gametogenesis. Gametogenesis can be in animals or in plants, because both of them, they produce gametes. So this is gametogenesis, or is the process by which the gametes are formed. And of course, the meiosis is one stage of this gamete formation, as you knew before. Right. Let's begin by the gametogenesis in mammals. Not in all animals, but in mammals, that's enough. In mammals, we have, of course, male mammals, and I told you before that the males, they have gonads, and which are called the testes, of course, and a part, a group of cells in these testes, they are going to form the gametes. I explained this several times. While the female mammal, she has gonads, which are the ovaries, and some of these ovarian cells are going to give us the gametes. The cells, or these some cells, are called the primordial germ cells. If I have an ovary like this, all of the cells, they are 2n. So part of these cells only, they will form the ova, and these are the primordial germ cells, and they are as well 2n. For the testis, it is 2n, all the cells, but some cells, they will be responsible for, or they will be concerned with the production of sperms, and they are called the primordial germ cells. It means that these types of cells are found in both testis and ovary. But let's begin now by the formation of sperms, and this is called spermatogenesis. I'm going to put a picture for both formation of sperms and ova, but I'm going to explain them separately. And by the way, in the book, you will not find this drawing or this picture. It was found in the old Edexcel syllabus, Now, in the new one, in the book, they just explained it without a drawing. I kept the drawing, هتلاقوها in the notes, of course, when I send them, in order to know how the spermatogenesis happened. So this is spermatogenesis, or the formation of sperms. As I told you before, that some cells of the testis, they are called the primordial germ cells, and they are 2n. They are going to divide, during when these cells are going to form sperms, they have 2n number of chromosomes. طيب يا سلمى, they divided to give me 2n again. Which kind of division is this?
Miss, sorry to disturb, but I can't view the picture.
You cannot see it? Okay. Can you see it now?
Yes, I can see
Okay.
it. Thank you.
You're welcome. Now here, this is the spermatogenesis. And I said before that there are certain cells of the testis that are called primordial germ cells, and they are 2n. When they divide, which kind of division is this, ya Salma? Mitosis or meiosis?
Mitosis.
Excellent. Bravo, ya Salma. Mitosis. طيب كملي بقى يا سلمى. When each primordial germ cell divides again, which kind of division again?
It's also mitosis.
Also mitosis. Bravo عليك. يبقى this means that every primordial germ cell is going to divide by mitosis twice. The resulting offspring or the resulting daughter cell after the second division, they are called spermatogonia. Here is the word, maybe it's hazy, spermatogonia. And I'm going to take only one of them to complete with it, and the rest are going to complete as well. Okay, spermatogonia, we have 4 coming out of each primordial germ cell. The 4 of them are going to complete these steps. So I'm going to take one of them as an example. This spermatogonia is going to grow in size. Look at the size here and the size here. It grew in size. When it grows in size, it gives me a cell which is called the primary spermatocyte. Primary spermatocyte. So what's the meaning of spermatocyte? As I said before, that the cyte means cell. It's meaning spermatocell, but it is called spermatocyte. This spermatocyte is going to divide by meiosis, reduction, division, to produce 4 of them. In fact, when the primary spermatocytes divides by meiosis once, or at the first, بصوا هنا دي إيه? This is the first meiotic division, meiosis 1. Remember, when I explained meiosis, meiosis 1 resulted in 2 cells, but meiosis 2 resulted in 4 cells. For here, meiosis 1 will result in 2 cells, each one of them having half the number of chromosomes. And these cells, they are called secondary spermatocytes. The secondary spermatocytes will continue meiosis 2, which was mitosis بقى, to give me 4, but not, we do not call them spermatocytes anymore, 4 spermatids. Here is the word, spermatids. Okay, these spermatids are immature sperms. When they grow to be mature, they are going to give me the sperm cells. And by the way, the scientific name for sperm cell is called spermatozoa. These are all of them. One of them is called spermatozoon. One sperm, or sperm cell, its scientific name is spermatozoon. All of them spermatozoa. Let me repeat again. let me revise كده with you. I have here one cell of the testis that's going to give me the sperms. So it is called the primordial germ cell. Do I have only one? I have thousands, millions, whatever, of primordial germ cells. Each cell is going to divide twice by mitosis, resulting in 2 N cells as the mother cell, mitosis. After the second division, the daughter cells, they are called spermatogonia. Each spermatogonium is going to grow in size to give me the primary spermatocyte. Here starts meiosis. This primary spermatocyte will divide the first meiotic division to give me secondary spermatocytes. These are going to divide the second meiotic division to give me 4 spermatids. These spermatids will grow and mature to give me 4 sperms or 4 spermatozoa. Type. Of course, I have only, this is an example by one primordial germ cell. And I said there are thousands or even millions. But how many millions of sperms are going to be produced at the end? Millions and millions of sperms. And I'm going to tell you the number later on. But let's go to the female. And by the way, this process continues as from puberty till the end of life of the male. Spermatogenesis does not stop. It starts at puberty and continues till the end of life. But in the females, it's different. The process continues from puberty till the menopause. It means that there is an end to the process of formation of ova, and the process is called oogenesis. يعني ال sperms, they are called spermatogenesis. This is oogenesis. طب let's begin بقى from the girl since she was a fetus. When her reproductive organs are still developing, when she's a fetus inside the uterus of her mother, it starts when the baby is a fetus. Okay? When the ovaries are formed, part of the cells of the ovary, which are called primordial germ cells, they are going to continue to form the ova. But this is one primordial germ cells, and of course there are thousands inside the ovary, in the material of the ovary. During the fetal life, this primordial germ cell, again, exactly like this, they will divide twice. And by the second meiotic division, they are not called primordial germ cells anymore. They are called oogonia. One of them oogonium, and all of them oogonia. So one primordial germ cell is going to give me 4 oogonia having 2n number of chromosomes. This happens when the baby girl is still a fetus. It's not born yet. The oogonia stay like this until the girl reaches puberty. In puberty, each one of these is going to grow up in size to give me a primary oocyte, exactly like here. When the spermatogonia increased in size, it's a primary spermatocyte. Up till here, we have the same steps. And this is at puberty. Okay? Now, the girl is having an ovary, and part of the cells, of the ovarian cells, they are primary oogonia. At puberty, every month, every ovarian cycle, one of these primary oogonia is going to continue. Every month means every 28 days, every menstrual cycle. One of these these primary oocytes is going to divide by first meiotic division. First meiotic division, like here. But what's the difference between males and females? Look at the size. Here they are the same, means that all of them will have the same process later on, or will continue later on the same way. But in the female, no. When the primary oocyte divides, it gave me two cells. One of them is called the secondary oocyte, and it's large, having haploid number of chromosomes, while the other one is very, very small, and it is called the polar body. This polar body is going to degenerate. Either they degenerate from the beginning or they degenerate later. Yani they do not have any effect later on. The effect is for this secondary oocyte. At puberty, one primary oocyte, every menstrual cycle, it's going to divide by meiosis 1 to give me a secondary oocyte, and remains like this. If there is ovulation and there is a sperm, so this secondary oocyte is going to divide the second meiotic division. So here, meiosis 1. And if there is a sperm, بيبقى فيه, it continues as meiosis 2. But if there is no sperm, this secondary oocyte is going to be degenerated and menstruation happens. If there is fertilization or there is a sperm, this secondary oocyte is going to divide by meiosis again, our second meiotic division, to give me two cells again. One of them is the ovum that is going to be fertilized by the sperm, while the other one is another polar body. It means that it's going to degenerate. We do not need it. Polar bodies, here I have a polar body. Either this will die at the beginning or it can divide, but it's going to die with all of them. Polar bodies always degenerate, always. And we are left with only one ovum that is fertilized to give me the zygote. Well, by the way, when the sperm nucleus fuses with this nucleus, meiosis 2 happens. As soon as the sperm nucleus enters, meiosis 2 is going to continue. This is the formation or the gametogenesis in male and female mammals in general, not only humans. But why do we write 2N and N? Because I said for mammals. If for humans, I'm going to say 46 and 23. But this is for all the mammals. Right. Let me explain something about the sperm and the ovum. Sperms, or as we call them, spermatozoa. First, one of them is spermatozoon, all of them are spermatozoa. Let me describe them. We always have 3 M's. They are many, mini, motile. So why many? Because when the male ejaculates the semen, this, in every ejaculation, there is about from 20 to 40 millions of sperms. That's why I told you that one primordial germ cell is going out. The primordial germ cells are going to give me millions and millions of sperms. Actually, this number is not written in the book, but I know it. That I have to describe what's the meaning of many, because you're going to think they are tens or hundreds. No, they are millions, 20 to 40 millions of sperms in every ejaculation of semen from the men. Many, because they are microscopic, yani the size of the sperm, the whole sperm is 50 micrometers. 50 micrometers, I'm going to show you that most of it is a tail that is not needed. Why motile? Because they can move towards the ovum. They have to reach the ovum. But let me put a picture to the sperm and explain its parts. Look here. If you can see, tell you no need for zooming in. The picture is clear. This is 50 micrometers. But what about these parts? They are not needed. This is they are not needed in fertilization. This is the tail. This is the neck, or sometimes we call it the mid-piece or the neck of the sperm, and this is the head. This is the most important part. All this length is 50 micrometers. And most of it is not needed. It's going to degenerate. Here we have this head. This is the nucleus of the spermatozoa or the sperm that contains n number of chromosomes. Okay? At the beginning of the head, there is a structure that is called acrosome. This acrosome is a sac that contains digestive enzymes. And it's found at the front of the sperm like this. Now, what about the neck or the mid-piece? The mid-piece is full of mitochondria. All these are mitochondria. Remember when I said that we have highly active cells, we have less active, and we do not have inactive. A highly active cell, I give you an example, liver, muscle, brain, and sperm. The sperm is one of the highly active cells. That's why it contains a lot of mitochondria in the mid-piece or in the neck. But what about this? This is a tail that is exactly similar in structure to the flagellum of bacteria. Sometimes we do not call it a tail, we say that it is a flagellum because, as you can see here inside, it has microtubules like those of bacteria. Remember when I was explaining bacteria, I said that the flagellum is several microtubules that are coiled like this. This is the same as the tail of the sperm. And when this tail moves to and fro like this or sideways, it allows the sperms to be driven forward in a manner similar to the movement of bacteria. I told you that the movement of bacteria is like coiling like this or running in spiral form to be driven forward. The sperm has the tail similar to the structure of flagellum, and the way of movement, it is rotatory movement or rotating movement. It rotates very, very fast and is driven forward. But this is for the sperm. By the way, I said that there are millions of sperms in every ejaculation. Only one is going to fertilize the ovum. But what about the rest of the millions? They are degenerated, they die. We consider this as a biological wastage. I'm wasting a living organism. Okay, when this question comes in the test, it is very silly question. What's meant by biological wastage in sperms? We are going to say that there are millions produced and only one is going to fertilize the ovum and the rest degenerate. So we lose living organisms. Right. What else? طب let's go to the Excel بقى أو الأوفم. اشمعنى السبرم يعني. Let me put the Excel, where is it? أهي. Look at this. And I want to show you meiosis 1. أو this is طبعا the meiosis 2 هنا مش 1. طيب of course the egg cell or the ovum, if I want to describe it, I'm going to say that one ovum is I will describe them as few, 3 F's, fat, fixed, 3 F's. تانية كانت 3 M. طب why few? Because one of them only is going to be produced in ovulation. Ovulation means that one of the ova, it is extruded out of the ovary to reach the oviduct. But again, why few? Because as I said that the sperms are produced in millions, هنا female baby, she is born with thousands only. And these thousands are enough for all the ovarian cycles or the menstrual cycles throughout her life, because we know that ovulation or menstruation begins at puberty and ends at the menopause. If you looked at this word, meno means menses, and menses means menstruation. Okay, let it be menstruation, meno. Pose, as if you're posing something, stop. But it cannot resume again, of course. Why there is stopping of menstruation? Because the ovum finished. They were only thousands, and this was enough for thousands of menstrual cycles throughout the female's life. Type. This is few. What about fat? As you can see that it is much larger than the sperm. Yani the whole length of the sperm was 50 micrometers. The egg cell is 100 micrometers. Nearly double the size of the sperm. This is the size of the human egg cell or human ovum. Is the size of the ova the same in all mammals? No, it's not the same. It depends upon the mammal. For example, the ovum of the ostrich, it is the largest ovum in all animals in general. It is 6 millimeters diameter. Yani here it millimeters. This is 0.1 millimeter. If I changed it, look at this and look at this. You can see the ovum of the ostrich with your naked eye. It's more than half a centimeter. So then you can see it. Okay, the ovum, not the egg. Of course, the egg of the ostrich is very, very large. Right. Fixed and fat also, because look at the cytoplasm here. It's a great amount of cytoplasm. Why? Because this cytoplasm carries food reserves for the growing zygote. When the zygote is formed and it starts to divide, it has to gain nutrition. So this food or this nutrition is supplied or provided to the zygote from the stores in the cytoplasm. The amount of the cytoplasm أو thickness of the cytoplasm differs as well from one animal to another. يعني for example, in the birds and reptiles, the chick here, or the offspring of the reptiles, they stay inside the egg until they completely develop, and then they hatch and they get out. Where do they get food from? From the greatest or the larger amounts of cytoplasm inside their eggs, having more and more food reserves. Birds and reptiles' eggs, they have a great amount of cytoplasm and great amount of food reserves too, because the embryo inside the egg is developing completely until it gets out. طب why humans do not have this great amount of cytoplasm? We know that the fetus is completely developed when it is born, exactly like the bird and reptiles. They are completely developed when they hatch. That's why they must have a large amount of cytoplasm. But why not humans? They are the same. Or why not mammals? The baby is born fully developed. Why the amount of cytoplasm is less? Because when the sperm fertilizes an ovum and a zygote is formed, this zygote will depend on the food stores for a very short period of time, days, until it is embedded in the wall of the uterus. Then the placenta starts to form. Let me remind you about this. You learned this, I know, in OL. Let me draw the uterus of the mother like this. And here are the oviducts. Here is another one with these finger-like projections that catch the ovum when it jumps out of the ovary. This is the rest of the uterus. This is the cervix. This is the vagina. Now here, the ovum, when ovulation happens and it is extruded out or exploded out of the ovary, it is caught by these finger-like projections that allow the ovum to enter the oviduct. By the cilia that are found inside and the mucus, and I explained this in cystic fibrosis, so the egg cell is going to, or the ovum is going to move till here. Yani the ovum is not moving, it was driven or swept by the cilia. That's why I said it is fixed. It's not moving, does not have any movement organ, or flagellum, or tail like the sperm. No, it is fixed, but it is driven forward in the oviduct by the movement of the cilia. It stays here until a sperm comes. When a sperm comes at this part, there is fertilization. And as soon as fertilization happens, a zygote is formed. And as soon as the zygote is formed, it begins to divide, as I said yesterday. And this is an activity, of course, so it needs nutrition. It gains nutrition from the cytoplasm inside the zygote. Until, and this goes on for several days, until the ovum or the zygote, it moves to reach here. It is embedded in the wall of the uterus like this. And when it is embedded, it is called the embryo. It's not called the zygote anymore. Placenta is going to be formed, and I showed you the placenta when I was explaining the chorionic villus sampling. It is the connection between the mother and the baby, or the embryo, through which food and oxygen is going to reach the growing embryo. The zygote depends on the stored food in the cytoplasm for a few days, and then the placenta is formed. That's why the mammals, they do not need a great amount of cytoplasm or great amount of food reserves in the cytoplasm of the ovum. But the birds and reptiles, no, they do not have a placenta or connection to the mother. The mother bird or the mother reptile, they just lay the eggs and go away. There is no connection between them. Yes, the birds, they sit on them to incubate them or to give them certain warmth, not more. They do not give them any nutrition or any kind of support. No, the bird or the chick inside, it is growing and developing by itself, depending on the food reserves that are found in the cytoplasm. Now let's go back to the ovum. I said there are few And I said why. They are fat because they are larger than the sperms, and they are fixed, and I mentioned why. Now let's look at the structure of the ovum. This is the cell membrane. Now where is the nucleus? These are the chromosomes, okay? And during cell division, there is no nuclear envelope and no nucleolus. Here, this is a stage أو at the beginning of meiosis أو the anaphase of meiosis 2. It means that this this ovum is fertilized or is about to be fertilized. So meiosis 2 continued. As you can see, this is anaphase where the spindle fibers retracted or shortened, pulling the chromatids with them. Now, what is this? This is a jelly-like layer that protects the ovum. Is called, name is not written, zona pellucida. This is a jelly-like layer that protects the ovum. Now, what are these? These are extra protective cells. They are called follicular cells. Now, what is this? Remember the polar body that I told you it will die? This is the polar body. It's going to die. Okay, when I said that the primary oocyte, it divided to give me the secondary oocyte and a polar body. And I said this polar body is going to die. Yes, here is the polar body and it's going to die. It has nothing to do with the zygote and what's going to happen next. But this is for gametogenesis and the gametes of animals or mammals. What about the plants? Let me begin by gametogenesis in plants, or the formation of gametes in plants. The gametogenesis in plants is much complicated than in mammals. Why? Because it happens, or it takes place on two phases. The first phase is called the sporophyte generation. Sporophyte generation. And the second stage is called the gametophyte generation. But what's this and what's that? The sporophyte generation is that the diploid cells of the body cells, they are going to divide by meiosis to give me minute structures called the spores. Spores, they are very, very microscopic seeds. They look like seeds. If I germinated spores, they are going in fungi, they are going to give me a new fungus. The spore is a very, very minute structure. What about the gametophyte? The haploid cells, they are going to produce gametes, but by mitosis. And let me explain this back. Let us see this. But before I explain this, I'm going to tell you that in some plants, like for example, the mosses, the ferns, or in other words, the gymnosperms, the two phases, they are found on separate plants. On one plant, there is a sporophyte. On the other plant, there is a gametophyte. And this is not our concern. In flowering plants, which are the angiosperms, or the widely spread plants that we see, both phases, they happen on the same plant. The main plant produces the sporophyte generation, while the gametophyte generation is found on the gonads, which are the male gonads, called the anthers, and the female gonads are the ovaries, for example, as you know. But our concern is flowering plants, not non-flowering and not اللي هم gymnosperms. But let's go now to formation of pollens that are formed from the anthers, and I'd like to explain something at the beginning before I show you any picture. Let us revise together our OL work again about the flower. We have taken that if this is a flower, these are the petals or the colored part of the flower. We have here the female parts that look like a bottle like this. And here it is swollen. And these female parts are the stigma. And this neck is called the style. And this is the ovary. All the female parts, they are called the carpels. So this is one carpel. Most of the flowering plants, they have only one carpel. For example, the sunflower, it has many carpels. Okay, and it depends on the species of the plant. But what about the male parts? They are found like this. Here is a long thread, called the filament, that carries on top the gonad, which is the anther. And both of them, they are called the stamen. The male part is called the stamen, and it is formed of the anther, which is the gonad that's going to divide to give me the pollens or the gametes, and the filament is just to expose the anther outside the flower, طبعا for pollination and things like that. While the female parts, they are called the carpels, and they are formed of three parts: stigma, style, and ovary. I want to show you this anther because I'm going to begin by the formation of pollens, which is called, or the formation of the microgametes. Why micro? Because they are microscopic. You can never see a pollen with your naked eye. For most of the flowers, you can, if you take a part of the anther and you put it between two fingers like this, and you rubbed your fingers together, you're going to get out the pollens in the form of powder. This powder, one powder granule may contain hundreds of pollens. You cannot see them. That's why we call them the microgametes. Means very, very microscopic gametes. These are the pollens. Now look at this structure. This is the male stamen that I just drew. And the stamen is formed of two parts. This is the filament, or sometimes we call it the stalk, and this is the anther. So what about this drawing? I'm just taking the anther and a part of the stalk. This is the anther. As you can see that this anther is formed of four sacs. They are called the pollen sacs. So the pollens or the pollen grains are going to be found here, studded in these pollen sacs. I just wanted to show you this picture, and let's go to what happens so that the pollens are formed. Now, where is the picture? Microgametes, here we are, here it is. Where again, this picture is not found in the book. They drew only the pollen, but how the pollens are formed, they removed this picture again. Alhamdulillah, I kept them. I have a habit that when something is over, I delete it. I didn't. Thanks God. ساعدتني. It's going to help me now. يعني in the new book they just have this one only, okay? But this is the gametogenesis from the beginning. Now let's start. I said that the anther is formed of 4 pollen sacs, and they are going to produce the pollen grains, and these pollen grains are immature at the beginning, and then they will mature inside the pollen sacs as well. So in the pollen sac, there is a great amount of microspores, which are 2N, or diploid, or we call them the mother cells. They are body cells of the anther, okay? Here, these body cells, they are going to divide twice by meiosis. When I say twice, I mean first meiotic division and second meiotic division. It means that at the beginning, first meiotic division, it divided into 2 cells or microspores, each one of them having n, or half the number of this. Then each one of this will give me, by mitosis, which is meiosis 2, 4 at the end. And all of them, they have half the number of chromosomes to the mother microspore. All these are microspores as well, but they are haploid. And as we learned before, each cell gives 4 haploid cells. These cells are going to divide by mitosis to increase the amount of pollens. Yani, I just told you that 1 grain that you have in your hand, between your fingers, 1 grain of powder, it contains hundreds of pollens. It means that from every anther cell or pollen sac cell that produces 4, these 4 are going to divide by mitosis repeatedly. When they divide by mitosis, they are going to give me N or haploid cells. These haploid cells are going to grow to give me a pollen grain. Again, it's a microspore, because I told you that the word spore means a very, very minute structure, and it is microscopic as well. So all of them, they are called microspores. So if I want to describe the pollen grain, I say that it is a microspore. But look here. Our book, it just drew this, but the old book explained why one cell produced 2 nuclei like this. Because each cell, it's not included in the book, ya Jama. This cell is going to divide twice, such that the one like this, let me take one of these cells. When it divides, does not give 2 cells. It's going to give me 1 cell having 2 nuclei. As if the division happened inside the cell without separation of the 2 cells. Yani here, the cells are separated. But here, no, there is no separation. There is no cytokinesis. So this nucleus is going to divide twice, and both nuclei are found in the same pollen. Okay, this was again in the old book, not in the new. One of these nuclei is called the pollen tube nucleus, and the second one is called the generative nucleus. And surrounded by cytoplasm, and this is the cell membrane, and it is again surrounded by a protective layer that they call it the resistant wall, because it's protective. This wall differs in shape according to the species of the pollen grain. What about these nuclei? What are they going to do? Which one of them is going to fertilize the ovum? This one, the generative nucleus. What about this one? This one is responsible for formation of something called the pollen tube. And I'm going to explain it to you now and show you How does it happen? Now, this pollen is found in the pollen sac, and I say that it is a mature pollen. It is ready to be taken by an insect, by wind, by a bird, by any animal, by man. It is ready to be taken to be introduced to the female parts. And the process of introduction of the translocation of the pollen grain from the pollen sac to the female stigma, this process is called pollination. Pollination is just carrying or transferring the pollen grain from its place in the pollen sac to the stigma of the female to start the process of reproduction. When pollination happens and this pollen is found on the stigma of the female, this generative nucleus, which is this one, is going to divide once by mitosis. يعني هي كانت, it was N, it will give me 2 nuclei. These are the generative nuclei, okay, because one of them is going to divide into 2 and both of them are haploid. So what about the tube nucleus? Yes, it is still there. It didn't divide. So this is the tube nucleus, and this is the generative that is going to generate a new organism. This happens as soon as the pollen is found on the stigma of the female part of the organs of the female, I mean. Type. What about the formation of the ova in female parts? I'm going to explain the female parts, and then I'm going to mention fertilization, or how does it happen. But let's go here to this picture. Give me a second to search for it. Here it is. I told you before that because always the ova, they are larger than, or the female parts are all, gametes are always larger than the male gametes. So I'm going to call this ovum, our formation of the egg cells, the ova, they are called the mega gametes because they are very large compared to the pollens. Now I have here this cell. It is called the megaspore. Megaspore means a spore, but it is larger than that of the male. تانية كانت microspore. This is megaspore. This megaspore is going to divide, as you can see. It has, of course, 2N. Okay, so it's going to divide, as you can see, into 1N, and this is meiosis. Meiosis 1. Then meiosis 2, where 4 haploid cells are going to be produced. Now remember what I said about the female mammal. When one of them is going to continue and the rest will degenerate, the same happens here. We have this meiosis 1 and then meiosis 2, resulting in 4 cells haploid. 3 of them, they are going to degenerate. Degenerate means they're going to die. خلاص, they're as if they are not there. Now, what about this one? It's going to grow. At the beginning, as you can see, the size is growing. So this is a megaspore, and this is found inside what we call here the embryo sac. This happens, of course, in the ovaries, okay? And this a megaspore is included inside, because it is found in the embryo sac, we call it the young embryo sac. Or in other words, we can call it a megaspore. Yani either you say megaspore or a young embryo sac, because it contains the ovum, the nucleus inside that is going to give us the embryo later on. This megaspore is going to divide 3 times by mitosis. Now here it was only one nucleus, N. Then it gave me 2 nuclei, 2N. These 2 are going to divide. This is the first mitotic division. Second mitotic division, the 2 nuclei will give us 4. The third mitotic division will give us 8. Let us count these back. Let us count them. I'm going to zoom this a bit. Yes. 1, 2, 3, 4, 5, 6, 7, 8. Because I have here 4 nuclei that divide. كلهم N. All of them are haploid. Okay. By mitosis, they are going to give me 8. Now, what are these? What's that? Only this one is going to be fertilized. This is the female gamete or the ovum.